All right. Good. Okay. Hello, everyone. And so my talk today is about controlling protein function with magnetic fields. Oh, good. Okay. And I am from Nonfiction Labs, which is a new startup that we just created recently in January. And we are trying to look at protein function with magnetic fields as a whole and turning it towards translational applications, such as developing these magnetogenetics and magnetotherapeutics possibly. Okay, so the vision overall for Nonfiction Labs as of January 2025, when we first got started on this, is to combine all of these different parts of light, magnets, optogenetic tools, using light for different protein functions and different effects in cells, and combine that into some sort of therapeutic that we could use in patients and develop that into something that will actually work, which right now does not exist. Okay. So to start out looking at protein functions, we can have many different forces that as physicists you might try to manipulate in order to modulate these functions of proteins to get your whatever desired effect you might want. So since we are looking mainly at like therapeutics, some of the important points that really matter to us are that you can have some genetic targeting, some methods that you can use all the genetic tools that we have, that there's some sort of specificity to eliminate off-target effects and all of that. Spatial targeting, so making sure we're actually getting to the right part of a body. Temporal control, that we can control when this whatever device modulation is activated and be able to turn it off when we don't want it anymore. And as well as the big one is to make it non-invasive and still have good depth penetration. Some different forces that we might try to look at are, we'll move on. Oh, okay. Oh, well, here's a bunch of them, but to start out, we can think about electric fields, such as like the Neuralink, which is currently a device that is starting to be used in patients. And it has some spatial targeting and good temporal control for electric fields because we can manipulate those. But it's not exactly a genetic technique, so it doesn't have a lot of specificity, as well as it is not non-invasive, as you implant this into the brain, and there's a very technical surgery involved where you have to implant it. Then we can look at mechanical forces, but such as in these sonogenetic techniques. But the genetic techniques are sort of questionable in the specificity because a lot of them are things that can have effects over many different proteins, such as like thermal effects, and mechanical forces in general can affect a lot of proteins that are already existing in our bodies. So maybe not as good. Chemogenetics and chemical forces we can use, such as DREADs. And these give you those genetic abilities. And the great thing about this is you can just put a chemical inside of a person and it can have all of the penetration throughout them. But you don't get any of that spatial targeting, and temporal control is kind of hard to deal with inside of a person if you're just giving them a chemical. Light and using optogenetic tools, so activating things with light, gives us a lot of these different things that we might want. However, as we've talked about in previous talks, light is not very good at going through the human body. That's why you don't see through the back of me. And so getting some sort of mixture of these techniques so that we can get all of the things we want, but not have the negatives is sort of what we're looking at. Okay. So what about magnetic fields? That's the big question as another force that we haven't yet gone to. Oh, sure. Sorry. Okay, perfect. Yes. All right, so with magnetic fields, oh, skipped ahead. The questions, we know that we can get a lot of these with the nature of magnetic fields, right? So we have, we have definitely spatial targeting. As we know with like MRIs, you can have very good control of these magnetic fields and shape them. You can get temporal control because we can control these magnets, such as with electromagnets, and they're very good at penetrating through the body with relatively small fields. In addition, we have some sort of specificity because regular biology is pretty non-responsive to magnetic fields. It's actually very hard to find something that does respond, which is actually a bonus for us in making a therapeutic because we're not going to be messing with anything else. The only question is, can proteins actually respond at all to magnetic fields? Can we have any sort of thing that we could have with our genetic techniques that we could use with magnetic fields? And as we saw a lot of techniques with like nanodiamonds and different stuff, but we really want to go into a genetic technique to give us all of the advantages of drugs and other therapeutics. Okay, so we had this question, and we started by first, years ago, we were looking at magnetic fields and trying to mess around with a bunch of proteins and just see what happens. And as you know, people who work with fluorescent proteins a lot said, why don't we just throw some of these in the microscope and see what happens? Lucky for us, we found that actually many fluorescent proteins do respond to magnetic fields, and you can actually control the fluorescence emission with your magnetic fields. So here we're modulating the magnetic fields on and off and seeing how the fluorescence changes with that magnetic field. So really cool finding. And in addition, we looked here at GFP and M scarlet, but if you're interested, try throwing a fluorescent protein under the microscope because we've looked at a lot, and a lot of these fluorescent proteins actually do show these magnetic field responses. So they're oftentimes small. You have to make sure you're getting the right parameters, but a lot of them you can find them. Okay. So looking into these effects more, M scarlet has been, we've studied that more intensively, and with these magnetic fields we've found, and there's this great paper by Catherine Zhang in the Cohen lab at Harvard, where she really delves into the mechanism of how this is happening. And this is through a magnetic field effect is due to a radical pair mechanism with an FMN photoproduct. So an FMN is a necessary cofactor that's used with this scarlet, and together they have this radical pair mechanism like we heard about before. And that's how you can get this great control of the magnetic field, or the fluorescence with magnetic field, where we can get 20% change in fluorescence. Okay. So other proteins that we wanted to look at, we can go to such as the love domain. This is an important light responsive protein and is controlled by magnetic fields. As we see here, we found that. And the cool thing about this is that using these proteins gives us a lot of levers to pull on to try to get these magnetic field effects to be stronger and to enhance them. So proteins, as we learned about at the very beginning of this yesterday, come from DNA that's been translated. And so we can make small mutations to that DNA. So we can have little point mutations to slightly alter what the DNA will end up being, and then translate that into proteins. So we take a big library of lots of different small changes in the protein structure. We can put those into cells. So here we're looking at, in the middle is a plate that has a lot of different colonies of cells. Each one of those has a different point mutation in it. So a very slightly different structure of the protein that's inside of them. If we image across all of these different colonies of all these different cells, we can identify in each one of them what the behavior is of that protein. And then we can isolate which ones we want to look at and which ones have whatever effect we desire choose those ones, and then see what it comes out of it. So through this means we don't have to know anything about what the mechanism is or how the protein is doing it, what is necessary, and we can just naively screen through a lot of different random mutations or anything we might think might be interesting and find a new variant that gives us an increased effect. Okay, so using this, we were able to develop this magnetic sensitive love domain into this mag-love protein. So over many rounds of screening, we went initially from a 1% effect in the magnetic field effect of the love domain fluorescence to, at first, for the first mag-love variant, it was 75%. Through later rounds of screening and more generations, we've now gotten up to 300% change in fluorescence of the love domain with magnetic fields. So the original love domain, the mag-love, is on adenine. I put the number here. And the improved mag-love 2 mutants are used in this paper by Harrison Steele's lab. And so they have the sequences for that if you want to try our new and improved version. Okay, so just to quickly shout out, as we hopefully already heard from the previous talks before, is that what's really cool about these effects as well in both M-Scarlet as well as the mag-love is that they've been shown by both Sean Bird and Harrison Steele and Gabby to have these resonance effects. So that means we can use RF fields, we can control them much better with localization, and we have much more control over them. It's really neat. All right. So I'm gonna assume that not a lot of people here work with proteins extensively. And so the love domain is a really important protein that has been used in a lot of genetic engineering efforts. And that's because it has an important effect, which is that starting from its structure, it can have a conformational change, which is a change in its shape based on when it's affected by light. So it absorbs the light, you can stimulate it, and then it will change its shape. So you can see the little helix that then moves. You can then tie this to some downstream effect. So if you have some protein with some behavior of interest, you can then tie this to then affect the structure of that protein of interest, like a binder, and have that give you downstream effects to turn the binding of that on and off with light. So this has been used extensively in development of optogenetic tools, right? So such as in here, they're controlling motility with light. So in the little video, they're putting a little dot and they're having the cell crawl towards the light as the light induces these actin filaments to grow in the certain section of the cell. We can also have this love domain is used in these light switchable nanobodies, which is like basically a version of an antibody. And so that means you can control antibody binding with light. So that's optogenetic tools. And we've seen how we're controlling these things with fluorescence, and we're stimulating them with light and seeing changes with the magnetic field. But as someone had mentioned before in their question, that means now we're still using light. So we no longer have this non-invasive good depth penetration if you still have to use light in addition to magnetic fields. That creates a problem for us. So we have checked off the genetic targeting, but now we're not as sure on depth penetration. We're thinking, what can you do to produce your own light? Well, we know from biology all around us, such as if you've seen a firefly, that there are ways to produce light. So the mechanism of like fireflies and bioluminescence in general is that they have proteins called luciferases, which when a substrate is added, they can have this chemical change and then it will produce light. So now we can have our own light production inside of the cell, right? These luciferases have been used before And they've been used for activating these optogenetic tools even. But the issue so far has been you're basically choosing, do you want it to be a chemical technique where you're adding the substrate to activate the light, but now you have a chemical that you have to add? Or do you want to have light induced your effect? But our hope is that we can tie the magnetic field to this light production, and then we can combine that so that we can have all the effects that we can get with magnetic fields, and we can still get our light from this luciferase. All right. So here's the grand plan. We can have our luciferase controlled by magnets, and then we can combine that with optogenetic tools and get our modular magnetogenetics. So I want to show you here our new protein MagLuc, and that is a magnetically controllable luciferase. So here we've put our love domain into a luciferase protein and shown that the luminescence can change with magnetic field. So we don't exactly know the mechanism right now, in that is the luciferase having a conformational change, or is there just an electron transfer, something going on? We're not really sure. We know we can see an effect in the luminescence, but if anyone has any ideas of how to study what exactly is going on, we would love to hear about them. Okay. And in addition, we're trying to take this, and just as we did with MagLove, where we can change from a small starting effect and increase that by orders of magnitude, we'd love to do that with this luminescence to develop it into a tool that could be used for these modular magnetogenetics techniques. So we're starting to do that engineering right now, but our starting point for changes in luminescence is only around 5%. So we still have a ways to go. If you want to get this initial MagLuc and start trying to engineer it to be better, you're welcome to get it on AdGene soon. We'll be posting it. And as well as if you're interested in the future and using these tools, once we've developed it to have a larger response, then you can reach out to plasmids@nonfictionlaboratories.com, and we would be happy to let you know when we have a new plasmid and send it out to you. Okay. So just to quickly highlight, in addition to all of the things we're looking at, where we're looking at these therapeutics and very highly focusing on getting it to some translational state, there's still a huge amount of open research and other different things that can be used and done with these proteins. So as I think someone else had mentioned, but there's a lot of different things you can do with deep imaging and lock-in detection and all of those sorts of things that these proteins could be very useful for. Okay, so the last piece of the puzzle is we've also started looking now at these different optogenetic tools to combine our luminescence with an actual targetable downstream effect. And so looking at antibody binding here, we've studied in mammalian cells how we can use this luciferase and use that luminescence to target an antibody to bind on and off from the membrane by just adding the substrate pyrimidine. So if we can get this to be tied in with our magnetic field control of the luciferase, now we have magnetic field control inside the body. So what we've shown and what we want to develop is that we can control light with our magnetic field and then be able to tie that into all the existing techniques where light can be used to control downstream effects such as binding, which is used in different therapeutics. So this binding can be used in lots of different therapies. There's many different antibody drugs that are out on the market, and tox and off-target effects are a really big issue. So being able to have this magnetic control of these drugs so that they can be specifically activated in one part would be really helpful. So we're working on developing this magnetotherapeutic and some sort of fun magnetogenetics where we can do this. and this is our team. So I have been really lucky to join this team at Nonfiction Labs. And we have here as well as Maria, who is our scientific leader, and Richard, the CEO, as well as our team in Argentina. And they've been really great to work with. So we would love to send you some plasmids or help you out to get started and look at all of these different things that we just don't have the time to look at. I'll also thank you to all of our other collaborators and people who have helped us out along the way and taken our offer of proteins. And it's been great working with everyone. Okay, so feel free to reach out if you have questions, suggestions, or whatever. We will try to get back to you in a timely manner.
Take some questions. We're doing well on time.
Great.
Hi, great talk. In the luminescence traces of these magnetosensitive fluorescent proteins, presumably you're switching on and off the magnetic field, and
Yep.
the same shape keeps occurring in all these traces. Do you understand where that's coming from? Is it coming from a property of the protein or of the fluorescent protein, or is it coming from the instrument?
You mean like what the
there's
time
this
is?
negative curvature
Yeah.
ramp up and then a quick ramp down.
So yeah, so that's not due to our instrument. There is some time that it takes for our magnetic field to turn on and off, but overall these effects are much slower than that. So I would say it's definitely due to the protein. If you look at, we haven't studied it very much for the luminescence, but I know for the fluorescent proteins such as M scarlet, in Catherine's paper, she does a deep analysis of what determines those different rates. And I don't remember off the top of my head, but there's a bunch of different things that determine what those rates are.
I have two questions. First is the simpler one. So how much light do you actually need to do the optogenetics?
It completely depends on the tool. So there's some tools that have been engineered to be extremely sensitive to light. And so they have very low needs in terms of intensity. And that's probably what we would do. And the thing is that even tools that currently require more light, there's a lot of room for protein engineering to get them lower. So common luminescence from these luciferases, just having a fusion protein of a luciferase with the optogenetic tool is enough to completely have enough range of intensity to modulate it.
Second question is when you use the fluorescence protein, how do you move it to where you want it to be? How does it get to where you want it?
So you're saying in like a therapeutics or just in general? It's non-invasive, so you can't just place it where you want it to be. Sure.
So how does it get there?
Yeah. So that's the part of the goal with this is to be able to just give someone a dose of whatever protein, and then as it goes through the whole body, so you would have it circulate, and it doesn't matter if it's in a different part of the body because it would be completely non-active when it's not in the region of interest that you're activating with your magnet.
So I may have missed the detail on this. How big are these fields that you're applying? Is this like a EMS pulse, or is this?
Yeah, I didn't mention, but relatively low. So we've been just using strong field, strong like fridge magnet basically. So I think that it's around 10 millitesla.
Wait, so is this like a neodymium magnet? You just
Yep. That's exactly what we use.
Easily, yeah.
Yep. So the idea is not to have huge field strength that's needed, so that it would be an accessible technique that you don't have to be in an MRI machine. You don't have to be in a huge magnet. You could just carry it a small magnet around with you that's attached to your chest or something.
If John sets up, maybe I'll ask the last question. Oh, sorry. Yeah, I wanted to ask about luciferase case, okay?
Uh-huh.
This, of course, produces light.
Yep.
But it's one photon per, no, one photon per molecule that you provide. So you burn those molecules.
Yep.
So the question is, how do you envision? Do you envision localized injection? Do you envision, is it toxic? How much
Sure.
can you take? Do you have chemistry throughout the entire body?
That's a lot of the questions that we're working on answering. I think right now the answer is that it would probably be some sort of like systemic dose or something like that that you would have to keep taking to keep it activated, something like that. And then it could just go throughout the whole body. So
It's not toxic.
as we know, it's not too toxic. But that's something we would work on and have to engineer around for sure. But oh,
All right,
did you have?
thank the speaker and